Hello, welcome back to Drugs and the Brain on Coursera. The next two modules are going to be dealing with receptors and the fact that they're proteins. So let's get started with a couple of concepts. First of all, a receptor. Simply enough, a receptor is a molecule on the cell surface or sometimes in the cell interior that has an affinity for a specific molecule, the ligand. Now, the ligand comes from the Latin word for something that we tie, and the ligand does indeed make a connection, a non-covalent connection with the protein in order to activate the receptor protein. The other concept, of course, is that most drug receptors are proteins. The word protein itself arose in the late 19th century when biochemists discovered these classes of molecules and correctly concluded that they are the most, or one of the most important molecules in the body, of first importance, and so they named them after the Greek word for first. Those concepts now we have in hand, and let's go on to the basic makeup of a protein. Here in a protein we see an amide bond or a peptide bond. It has a carbonyl oxygen and it has an amino hydrogen. So this is the backbone of a protein, and at one end of the protein we have a string of these amide or peptide bonds. At one end we have an unbound amino group. That's the amino terminal end of the polypeptide chain. So we have many peptide bonds together. We call it a polypeptide. At the other end, we have the carboxyl terminal end of the polypeptide chain, a carboxyl group that's not associated in a peptide bond. So that's our polypeptide chain, and the peptide or amide bonds link the backbone or main chain or alpha carbons. Here's an alpha carbon, here's an alpha carbon, here's an alpha carbon, and every protein has a series of these amide bonds and a series of alpha carbons. The reason that we begin with the N terminal and then go to the C terminal is that that's how the cell makes the protein. First the N terminal, then adds other amino acid residues onto the C terminal. An amino acid literally has an amino group, an acid group, and a side chain. And it is, of course, the side chains of each amino acid residue that do the chemistry of the protein. So the side chains can be aromatic, they can be hydroxyls, they can be carboxylates, they can be acidic groups. There are a total of 20 types of amino acid side chains, and they are responsible, again, for the chemistry and the folding and most aspects of the protein. We won't need to learn the names of the side chains, and we won't need to learn the chemistry of the side chains in this course. The shortest peptide or protein in the nervous system has only 9 amino acids, and the longest has 5,500 amino acids. So there's a tremendous range in the characteristics of proteins in the body and of proteins in the nervous system. And again, virtually all receptors are proteins. Now, proteins contain a few structural motifs, a few basic kinds of putting together strings of amino acids, and I will show you those structural motifs right now. The program that we're using is called Swiss PDB Viewer. There are several programs available. The file that I've used is a PDB file that I have constructed myself. It's not really a protein that exists in the structure database, not really a protein that you would find in nature. It happens to be simply all 20 amino acids strung together to show you the principle of the alpha helix. The alpha helix was described first in the late 40s by Linus Pauling right here at Caltech, and I'd like to show you several aspects of the alpha helix. Here I'm rotating it around to show you the backbone and to give you a feeling for the three-dimensional aspects. The first thing that we're going to do is that we're going to bring up this control panel here, and we will hide the side chains. There we are. So now we're showing just the main chain, or the alpha carbons. You can see that it's a very nice helical structure. We'll bring the side chains, we'll keep the side chains hidden. The next important aspect of a protein is that we are going to show you the hydrogen bonds. Oh, let's see, we'll have to display the H bonds. There we are. So the hydrogen bonds go from a carbonyl group to an NH group, one turn up in the alpha helix, and they are the non-covalent bonds that hold the alpha helix together. And since they're regular and staggered, they do a good job of holding the alpha helix together if the side chains will allow this. So let's put the side chains back. And the next manipulation will be to show you the ribbons, this abstraction that does not really exist in the protein, but clearly demonstrates the alpha helix. The ribbon has an arrow at one end toward the amino terminus. So we've showed you the ribbons and the arrows. Let's see if we can show you the van der Waals radii, which is actually, we'll turn off the ribbon here, which is actually the extent of the atoms. So here we have the van der Waals radii, which is what would be experienced by an atom in the vicinity. Okay, now we'll show you a stereo view. And so we'll show you stereo. Okay, now if you, to see this in stereo, what we need to do is to practice with putting our eyes together. I'm making it a little easier for myself to do this. And after a while, you can focus. And now, as we rotate the protein around, you can actually see it rotating in 3 dimensions, which is a very nice way to get a view of the 3-dimensional aspect of the protein. Okay, and you can manipulate these proteins for yourself. It's probably not so interesting to do it with this fake, very small portion. In the next module, we'll show you a real protein. You can download that structure and you can go to the database. The next model to show you will be a beta sheet. So we'll bring the beta sheet onto the screen, and we'll make it a little bit larger. And let's put the control panel on. Window, control panel, there it is. And let's have a look at this beta sheet. Now, there are 2 beta strands, anti-parallel. Again, we have just used an alphabetical order of side chains. And we'll show you the hydrogen bonds again. Tools, compute H bonds. Now we see that the hydrogen bonds actually exist between the 2 chains rather than within a chain, and that's what holds a beta sheet so stable. Here we are, we can rotate it around. They are between the alpha, again between the carbonyl group and an amino group, but in this case now it's between the 2 chains. And we can abbreviate a beta strand again by a ribbon structure, and here are those 2 straight arrows. You see an arrowhead up here and an arrowhead down here, so that is the abbreviation or the abstraction of a beta strand, and these 2 beta strands together form a part of a beta sheet. So we've seen the side chains. We can show the van der Waals radii again in this structure, and so this is actually the space-filling version. We see that the 2 strands are actually quite close together, interacting with each other quite nicely. And we can also do a stereo view. Let's go back to the simple view, and we will turn off the ribbon and look again at the stereo view. So we'll do display stereo view, and we'll bring them a bit closer together so that we don't have to have our eyes so far apart, because in a stereo view you do like to have the eyes fairly close together. And we'll bring it up to be full screen. Let's see if we can now put our eyes together and see the stereo. Well, I can see it, and I hope that you can also. And we have a nice view of the protein rotating around in 3 dimensions. You can do this on your own screen when you download various structure files. All right, so we have shown you the basic parts of a protein, and next time we'll do a little more about receptors as proteins.